All right, it's got dark. So this is kind of a quick, that's not going to be too distracting. There you go. Keep my head in one place. Sorry, dog's got a new squeaky toy. God. All right, let's start again. Try and ignore squeaky toy. All right, so this is just a kind of going over of the two assignments tied to the Fly linkage project. First of those is an Excel spreadsheet, which essentially contains your data analysis. Right, your data analysis consists of two kind of broad categories. One is visual representation of your data, right? Because what I've posted on Blackboard, the picture of the whiteboard from the lab, obviously you can get a pretty good idea of, you know, what's going on just by looking at that, because the numbers are pretty extreme. But it's not the best way to interact with that information, right? So the standard is a graph, right? And how you do that really depends on what story you want to tell. So there are many different ways in which you can graph a set of data. It simply depends on what you want to use that graph to say, essentially, right? I mean, essentially, when you're writing a paper, even if it's a lab report for genetics, you're still telling a story, right? And so if you think of it from that point of view, then it kind of makes a little bit more sense. Okay, so that's the first part. Share my screen because I want to actually show you some examples, right? The second part is kind of crunching those data, right? And so really, you want to first see, are these two genes linked, right? Is something funky going on? For that, we'll use the odds ratio. Let me find, here we go. Yeah, this is the one, right? And all of what I'm kind of talking to you about is explained in a fabulously thorough video by Dr. Page, right? Which you can access from this link here. And it's so good, I've never felt the need to do it again myself, right? Because he does a very, very good job. And he uses this data set here. Obviously, you will use your own, right? That's very important. But you can work through all the steps using this data set along with him if you wish, right? And to be honest, as long as you follow all the steps in that video for your data, and you send me the Excel file with all of that in it, 100 points, right? Not hard. Takes a little bit of time, but it's also worth 1/8 of your lab grade too. So it's worth putting in that time because it will make a big difference. Okay, so graphing first, right? So you need to generate that graph. And again, it depends on, I don't know what's going on there, you want to say, right? So this, kind of weirdly, I might have moved it. No, it's just plain silly buggers. All right, so this is a graph of essentially parentals versus recombinants, right? And so if these two genes are not linked, and if we did the experiment properly, which we didn't, but that's another question entirely, you'd expect equal numbers of parentals and recombinants, right? So essentially, these are your expected values. This is what Dr. Page takes you through. And these are your actual values, right? These are what you observe, right? So clearly, these two things are very different, right? So what I mean by telling a story visually is you look at this and you can figure it out yourself, right? It's not very hard. It's kind of right there in front of you. So that's one way of telling that story. It's not the only way, because this is from, I can't remember who this was. Oh yeah, there you go. You can see the name up there. Let's kind of see. Get out of it. There you go. That makes it a little bit better. Right. And so the way that she, whoops, sorry. Get back over there. The way that she wanted to tell that story is by plotting the data by phenotype combination, right? So white-eyed yellow body, red-eyed wild-type body or brown-bodied, right? So these are the parentals, and these are the recombinant phenotypes, right? She has split it by sex, so she's essentially plotted all of the data. That's cool. You could also do a similar thing to the other graph. This one, I don't know what's going on with this. Anyway, where you plot the 4 different phenotypes, so you kind of smush your different sexes together. So you just give the phenotype number of that phenotype, male or female. And then you could do the number of expected, right? So for each of these, so instead of having 2 columns both of observed data, you could have the same as the other graph, where you have 1 column of observed and 1 column of expected for each of those 4 phenotype combinations. Any of those things work, right? It just depends on what you want to say, like, about those data. So that's the first thing that you need to do. The second is kind of a rough and ready, back of a napkin kind of statistical test to see if you're getting what you should expect, right? And that's called the odds ratio, right? Which is explained here, right? And so essentially this is just a simple 2x2 table, right? Where you put at the kind of where they meet is the combination of different alleles, right? So this square cell A is the number of double homozygous, well, double recessive. There you go. Let's talk about phenotypes, it's easier. Cell D is double dominant, right? Because they're plus plus. And then the other 2 cells are recombinants, right? Because they have 1 dominant, 1 recessive allele. And so quite simply, the odds ratio is the 2 parents times together divided by the 2 recombinants multiplied together, as you can see here, right? And essentially, the odds ratio, what it tells you is that if you get an equal number of everything, your odds ratio should be 1. And so if you get an equal number of each of the parentals and each of the recombinants, i.e. those 2 genes are not linked, right, because you have independent assortment, you should get an odds ratio of 1, right? And as you can see, the odds ratio here is basically 1 in 25,000 more likely to get these 2 things together or these 2 things together than to have some other random combination, i.e. something's going on. Now you want to test that a bit more rigorously, right? So now we do a chi-square test, and you should all be perfectly familiar with this. You can do it in different ways, right? And it also depends on your hypothesis, right? So a good working hypothesis is that there is no linkage between the white and the yellow genes. There's no linkage, then you expect an equal number of your 4 different phenotype combinations. So essentially your expected for each of those phenotype combinations is your total, whatever that might be, I haven't actually added them up, divided by 4. What Dr. Page has done it by sex, right? So essentially you have 8 different categories. You have the 4 different phenotype combinations for each sex. That's fine. You then just divide the total observed by 8. So in this case, I think it's 1,000, right? So 1,000 divided by 8 is 125. Easy peasy. I don't know what your total is for the lab, and obviously it will vary for each lab section. And then you just calculate the chi-squared value using your observed, using the formula that you see in front of you and the one that you've used a bunch of times already, and then use the chi-squared table and your degrees of freedom, however many that may be. In this case, it's going to be 7 because there's 8 categories. If you collate the sexes together and just look at phenotype categories, then it's degrees of freedom 4 categories, 3 degrees of freedom of 3. If you just do parentals versus recombinants, which is a bit of a blunt instrument, to be honest, I wouldn't do it that way, but you can, then you have a degrees of freedom of 1, right? So whichever way you do it, it's up to you. You need to present a p-value, right? So that's what we have here. So actually, the student did do it as parentals versus recombinants, which is kind of weird because you plotted it in a different way, but I don't know, it doesn't seem kind of consistent, but it doesn't matter, right? Because it again depends on what you want to say. And then finally, you want to work out the map distance. Now, this is going to be a little bit weird because our data are crazy. But still do the exercise anyway, right? And so from your lecture stuff, that's not a very good way of putting it, you'll know that recombination frequency is simply the number of recombinants divided by over the total number of observed, right? And whatever that is in percent is your distance in centimorgans, right? So one cool thing is, at least from the first section, because I haven't done that ones yet, is clearly recombinants are rare. They are present, but they are rare. So even though our data are weird and wacky, they still more or less fit what we expect with the, extra wackiness added on, right? Those excessive wild types that we've talked about before. So all of this, graph, chi-squared, odds ratio, map distance, all of that, save it in an Excel file with your name, or send me a link to your whatever this is, like online Excel, doesn't really matter. That's your Excel assignment, 100 points, easy peasy, right? And can quite quickly and easily tell you the student got 100 points, right? Not difficult at all. You do need to have your axes labeled, a key, so I know what I'm looking at. I can figure it out, but it'd be nice to be told. And that's pretty much it, right? Okay, so I think this yeah, so essentially the same, right? So this whole bit of a spelling mistake, but that's fine. I don't know why that conclusion was made because that's wrong. It doesn't matter. This is correct, right? So this student, whoever that was, also got 100 points, right? So that's the Excel spreadsheet. Check the schedule for when that is due. If you need a hand with any of it, give me a shout. More than happy to help. Now, the lab report is basically taking all of this and dunk into a more formal written version, right? And essentially what I, where is that? Here we go. Expect is the formal layout of a scientific report, right? As I've told others, actually, where would I find that? Don't want to see various pictures of my kids. I should know my own way around my computer. You would have thought. Oh, here we go. This is the most recent. Little paper. There's a chicken as well. All right, so this is kind of an equivalent of what you're doing, right? Except this has been published, right? So you can actually go look this up and read it if you wish. So essentially we have an abstract. You don't need to worry about an abstract because it's a fairly simple lab report and you can cover most of this in your intro anyway, right? Scroll down a bit, scroll. Okay, so essentially, this is a little bit different format, but it contains the same stuff. This format is dictated by the journal, essentially, right? Because these are notes, these are microcups, small publications. So first two paragraphs, introduction, right? This is what I work on, this is the gene I'm interested in, and this is what I'm looking at, right? Essentially, that's what the introduction is. Background on what you would need to know, right? And reviewers often go, hang on, you need to add that because I don't understand it, right? So you need to add enough that your audience understands what you're doing, right? You can't assume that they know all about Drosophila white and yellow genes, or in my case, egg-laying defects. So you have to give them that information. In your case, independent assortment, linkage, Mendel, Drosophila, stuff like that. For example, oh no, that's wrong. Wrong one. There we go. There you go. There's also a rubric in Blackboard in the Fly project folder. That is what I use. I copy, paste it into your lab report, and then I fill it out. Okay, so look at that and it'll tell you exactly what you need to do. Then you need to describe what you're doing, right? So that anyone reading this can follow along and if necessary, repeat it as well. That's essentially the gold standard. So this is a bit weird because I include kind of results and methods and results together, which I don't want you to do. What I want you to do is to talk about methods, which essentially is kind of further up in this document, right? It's not hard, but I do expect to have a brief description of how you do the cross. You can copy those figures, no issues with that whatsoever. And the analyses you're going to do. So essentially, you're going to tell me what you've already done in your Excel spreadsheet. Okay, easy peasy, free money, basically. Then the main important part, and this is the core of any scientific publication, are the results. These are the rock upon which you build your argument about what is going on. Can't do without that, right? And so the results I want from you are essentially all of what you did in your Excel spreadsheet. However, you also need a couple of extra things. One, you need a figure legend. What's the figure legend, you might say? Figure legend is, where would I do that? Here we go. This. So this is a little bit more complicated than what you need to do because the graph is a little bit more complicated. It's a different kind of deal, but the same principle applies. What are you showing me? I'm showing you two graphs. This is proportion of essentially normal worms, non-egg-laying defective worms, for a variety of different genotypes and statistical analysis, right? Normal, not normal, normal, not normal, basically. B, normal, 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 not normal, not normal, right? That's essentially what I'm saying. Obviously, a little bit more technical than that, right? And so you need to describe what is in your graph. And the aim of the game is you could take that figure legend and these figures out, and someone would be able to understand what they mean, like a standalone deal, right? So you need your figure legend. Obviously, the more figures you have, the more important this is. And also, you will be labeling them figure 1, figure 2, figure 3. So if you have like a nice cross diagram, where are we at? Or slide pictures or whatever, right? You can do whatever you like. You know, it helps you explain what you want to say, right? If you had put this in, this was your first one. This would be figure 1. If you put in another one, this will be figure 2. For your graph, let's see, example. No, that's not there. Crab apples. I thought I opened one. Oh yeah, here we go. Oh, unfortunately, this student didn't, right? So you get points knocked off, right? So she would have lost half of the points for her graph, right? 10 points for the graph. Well, sorry, 10 points for the graph and the figure legend, 5 for each. Don't put one in, don't get the points. So that was actually a kind of funny example. I did actually spend a little time looking, but my files are a little disorganized on this stuff. And you also need to add some text describing those results. Doesn't have to be a lot. I found that there was a vast excess of parentals, figure 1, right? Much higher than what I'd expect if there was no linkage, right? That's a description of your graph. It may seem a little redundant that you're doing both a description in the body of the results and a figure legend, but that's just kind of how it works. It makes more sense when you have more to talk about, essentially, with regards to a figure. So for example, talking about figure 1A here, right? So here are the results, right? So we found such and such and such and such, figure 1A. Hello, love. But when we looked at something else, we found no effects. That's figure 1A. Right? You need to do the same. Obviously, then I'll talk about figure 1B. So the results added together, 10 points for each of these things. Half of your grade for your mini lab report are your results. You don't put one of these in, don't get the points. Can't make it up elsewhere, right? Even if you write beautifully, you're not going to get extra points to make up for leaving something out of your results. And then you want a short discussion. Essentially, a discussion is summarizing your results, interpreting your results, and then summing it up. So, for example, so there's some nice text, right? Shown in figure 1, blah, blah. Great, it's exactly what you need. Right? And then I think the only thing that she didn't talk about here was the odds ratio, right? And she probably could add a little bit more. I think she probably got most of the points there for the discussion. So the equivalent in mine is this. Oops, get out of it. Right, not that bit, this bit. Do it the other way around. If I can. Whatever, you can see, last paragraph. Right, essentially, we've shown this, we've shown this, we've found this. It's different from something else we found in a different paper, and we've shown something new, right? This is really cool, even though we can't explain it. That's the discussion, right? So do all of that and write neatly, use paragraphs, you need headers. Again, look at the rubric. It's got everything there for you. And I follow that literally. It's a rubric, right? I will copy and paste that into your report and go through and have you got a discussion? Have you done this? This? Yeah, that's worth X points. Tick, 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 tick. Okay. So if you want to know how to get a good grade, look at the rubric. It'll tell you. All right. I think that's about it. Can't think of anything else off the top of my head. As always, the path to success and happiness, at least in terms of the genetics lab report, is ask for feedback. It's very rare that a student will do badly after giving me a rough draft and acting upon the suggestions that I make. Usually those students get great results, not results, grades, okay? And this is worth 1/4 of your lab grade, which is equal to, what's a quarter of a quarter? 16? I don't know. That's making my head hurt. Still a reasonable chunk of your overall course grade. So you really want to do well at this. All right, I think that's pretty much it. So as always, if you got questions, give me a shout. And also, really, truly, get me whatever you have, even if you haven't finished your lab report before the deadline, get me something. I'll give you feedback. It will be better. You'll get a better grade. All right. See you later.